So neuroscience technology, microscopy, and I will discuss about fluorescence microscope. And later on, even not light microscope, but electron microscope and how to process microscopic data. So who have not used fluorescence microscope before? Okay, who have not used fluorescence? Okay, good. Yeah, many of you haven't used, so this is a great chance. The first time you understand about fluorescence microscopy, we'll cover various things, epifluorescence, and very sophisticated technology called Confocal and Two-Photon laser scanning microscope. By itself is a semester of lectures. So if you can get the core idea, that would be excellent. And TILF is actually my PhD work, and I'll skip this due to the time limit. But later on, I'll be happy to talk with you if you have any questions on this. And the second part is electron microscopy. There, you guys heard we have many expensive microscopic facilities in our school, and we have a central research facility which has like $1 million, millions, tens of millions of this expensive equipment. It would be great if you know how they work and what you can expect if you have a sample. All right, so we will discuss about TEM and SEM, called the TEM and SEM in this class. And later, how to process image and how to interpret. We will discuss about this. All right, first of all, I want to discuss about fluorescence. In the old days, people realized they found a stone, okay, which has a special property. What kind of property it is? You shine, cellophane tape, like colored film. So you use this UV passing filter to shine this stone with the UV, and people realize that, hey, of course, if you shine some light, that will directly scatter and reflect, so you see the same color, of course. However, people realize, hey, there's some other colors are coming together. So to find it out, you actually put another cellophane tape, which is, let's say, blocking the original UV in here. It looks like purple, block, and then you still see some lights coming through. So can you imagine if you are looking through this, what happens? Except this stone, the rest of this light will be blocked. So the background will be complete black. And then the lights coming, the interesting light, which is here, looks like a blue light pass through. So you see a black sky, there's a blue things with this shape is coming into your eye. So that's the start. And that's the principle of this excitation light and emission light. The point is their wavelength. On the other hand, their energies are now different. And you can discriminate or distinguish the part, the molecule, or the substance, which we call as a fluorophore or fluorochrome, distinctly, and you can only watch this one. Okay, so how can you watch this? And let's think about the spectrum. So let's say this specific stone, or called the fluorochrome, chrome means color. So what's the property of this chrome? So this guy has a special absorption with respect to color. So let's take a look that we call in chemistry, we call it as absorption spectrum. So this stone somehow absorbs very well at the later, like 470 nanometer, let's say. It absorbs very well. It absorbs a little bit other spectrum, but there's a peak. And what happens? This absorption, in terms of quantum mechanical point of view, they absorb this energy of photon and they excite their energy state, and they actually use up. Some of them are leaving as in heat, or internally they get to the point, they use up some energy, and then they emit the energy as in the form of photon, and that we call emission or fluorescence emission. Look at what kind of the energy or spectrum distribution of emission in this case is. That looks like this. So it emits, it absorbs at the blue color, and it emits in the green color at the maximum. Of course, there's some overlay, which makes our life a little bit more difficult. However, the important thing is, you see the x-axis wavelength, the wavelength has been longer. So what you can do is you have put a filter and block out all this lower wavelength. Then you only see the fluorescence emission. You only see through the stone only, not the background, which is not our interest. Okay, this is the principle of fluorescence microscope. Why is it cool? If you look at Nature Science covers, many of the time you will look at black background and beautiful colors showing up. Those are fluorescence microscope. And this allows us to specifically look at this structure or molecule. And that's the key, which is provides a specific contrast for the fluorophore or fluorescent molecules. Okay, so specialized molecules, fluorophore is that absorbs light at specific wavelength and emitting light at different wavelengths. And typically, 유승영?
Yes.
Yeah.
Longer?
Yes, exactly. Long. 승영, one more. What's the energy of the longer wavelength photon compared to shorter wavelength? Longer wavelength, is energy higher or lower? This emission
Lower.
photon.
Lower?
Great, yes. So everyone should understand longer wavelength has a lower energy because the incident photon has higher energy. You somehow lose energy, and so you have a lower energy, which typically have a longer wavelength. You can simply consider, hey, you don't want to go into the sunlight, UV light. UV light has a shorter wavelength. It has a high energy. It's likely to damage our DNA in our skin to cause skin cancer and you know, pigmentation of our skin, which is not good for our aesthetics, right? So that's the concept of excitation photon and emission photon, which have different color or different energy. All right, so how can we utilize this for our biomedical or neuroscience research? You have this molecule, you attach this one to the structure of our interest, such as a neuronal cell or specific proteins only expressed in neuron, you attach this one, then you only watch this. You're not watching this thing, but you're watching this one fluorescence, okay? So it's a surrogate marker. All right, so that you can link to antibodies or other molecular probes to indicate presence of specific proteins or organelles. All right, so now let's design a fluorescence microscope, okay? So how can we design? How can you build a fluorescence microscope? In fact, it's not that difficult. You just need a microscope and somehow use filter to block out unnecessary light and to send only the light you want to shine onto the sample. So how? So let's say we have an incident light source. This light source typically can be a mercury arc lamp in the past. Now mercury is banned in the old hospital and many laboratories. So now we are using, for example, like LED light source or various light source which contains a various wavelength. But as you saw, this molecule is a specific. It only accepts a specific color. So you have to match that by using filter. In this case, this filter allows only pass this blue light. okay? Usually high energy or shorter wavelength. So that light has to shine onto your specimen, right? Then the other part which is shined, they don't do anything. They just scatter the light. But this specific fluorophore will emit another color of light, which is shown here as a dim red color. So that red color is emitting out. In fact, the emission goes out throughout every direction, in fact. But this one only depicts those who are entering into the microscope because that's only we can see and we care, right? So this red color is coming out. Now the issue is you have to capture this red color by using imaging device or microscope. Here you can see this lens. So lens focuses down this light coming out of the focus, a point, and it actually sent through either your eye or camera. So to make this possible, you actually have to invent a specific, very weird thing over here. What does that mean? This specific mirror has to reflect blue light while it has to pass through reddish color. So you need to invent this one to make this possible. So how can we name this one? Hey, two different color, we will have a different behavior. So one color will work like a mirror. The other color, it works like a window. So that's why I name it as a dichroic mirror. Di means two, chroic means color. So it functions differently. Okay, with the invention of this dichroic mirror, we now can pass this red color, but this is usually not perfect. That's why we also have another filter to block out all the scattered light from this excitation. So we need to have another barrier filter, and that goes through. So now we invented a fluorescence microscope. We designed it, and how to realize this? You can see this key component is a dichroic mirror, and excitation filter, and barrier filter. So these three units, you can put it together into filter cube. You can buy it about $100,000, $1,000 almost, okay? I have to say all these optics, fluorescence microscope components are precision machine, and it's typically very expensive. You have to understand that. So that's we call fluorescent filters. So look at this dichroic mirror, excitation, barrier filter. These three together, you can make it as a one cube. So look at this, light from this excitation filter, and this is dichroic, or we call dichromatic mirror, and then the light goes down, and then it reflects back, especially fluorescence light can pass through, and it has to pass through another filter called the barrier filter, or we call it as a emission filter. So this has 1, 2, 3 units inside, and that we call fluorescence filter block or cube. Does that make sense? So this is what happens when you are working in the laboratory, especially with a fluorescent protein or fluorescent molecules, to make an image of the distribution or the dynamic shape changes of the cells. Okay, so then how you have to know the characteristics of the filter. So just don't believe what you buy from the vendor, and just don't trust. You have to watch their specification. In this case, the spectrum. So I'm going to go through a little bit. This is a little bit hard, but it would be great if you understand this. So first of all, excitation filter, you want to narrow down only the color you want, okay? Especially lower wavelength. So in this case, you see, let's say this red one, the y-axis is transmission. So you transmit only this narrow part of this red colored part, so that you limit the excitation light. And then what happens? For the emission, you need to open up all the light, as much of photons as possible, but with a longer wavelength, as I said. So that is this blue emission filter, which you can see the transmission is high here, but transmission is almost very low on this background or lower wavelength. So that's key. In between, there's a dichromatic mirror, and you can see what this mirror does is actually block, actually not block. It's a transmission is 0 here, but transmission is high along this emission wavelength. But this is a little different from the other filter. The other filters only block the out of the wavelength, but the dichroic mirror has a mirror in it. What does that mean is it's different. Why? If the transmission is low, it doesn't block the light, it becomes a mirror. So this is a reflection for this green dichroic mirror. So this is reflection, it's not absorption or blocking. So that's the difference. Let me go back to give you one term which I want to emphasize. So this phenomenon of excitation light and emission light peak wavelength difference is discovered by Stokes. So Stokes put his name on here. So the tendency of this fluorophore to be excited and emit a longer wavelength is called Stokes shift. So you better to know this terminology. Okay, so now as I explained everything, okay, so you can put your questions anytime in chat. You know, this is not easy concept, so you can put your questions over here. Okay. So now I want to go a little bit deep inside anatomy of key parts in fluorescence microscope. Now you know how to make a fluorescence microscope, the key component of filter cube, but that filter cube can be a little bit different in different kind of microscopes. So here I have an example that this is a showing that emission light, excitation light. So the dichroic mirror works as a mirror. So it's in this case, green light gets in, it works as a mirror, so it gets down, shine onto the sample. So that's called excitation. And what about emission light? Now it's shown as a red. Emission light is a fluorescence, and that emission comes back, and now dichroic mirror has to transmit this. Okay, and then so that you can collect it to form a fluorescent images. And all this together looks like this. And if you have inside your lab or in our department, we have a fluorescence microscope. This is a anatomy of fluorescence microscope. And one thing you have to notice is this objective lens is not a single lens. You can see there are multiple lenses. That's why this is the most expensive part of your microscope. So never drop it. You have to work it like a diamond, like something. So you have to be very, very careful when you deal with this, okay? Okay, so now we have understanding of fluorescence microscope, but I have to give you a little bit of, in this situation, you have specimen, you notice that we shine the light from top to bottom above the sample, and we take image on top. So we call this as an epifluorescence microscope. Epi means on the surface, okay? So you are looking at shining from the top. Then what about the other kind? So there's a sample, you shine on top and you observe one from the top. So that's epifluorescence. The other way is you shine on the bottom, pass through the sample, and you observe from the top. That's called trans-illumination or transfluorescence. While unless the sample is very thin, like cells, you don't typically use trans-illumination much. So that's why epifluorescence is more popular in this case. And of course, this is a wide field microscope, which means you use a CCD or image sensor, and you're taking the whole picture in one time. So it's a shot, like your cell phone camera, okay? So what are the elements of this? You do need a specimen, and you do need excitation light source, and you need, I already said, dichroic or dichromatic mirror here. That makes this epifluorescence possible. So this is a magic for that. And you have emission light coming back. So with this, we have to consider what are the limitations of this approach. In fact, I have to say dichroic mirror and filters are not perfect. Of course, these days you pay more money, you have a better filter. But in general, dichroic mirror is not perfect. Let me show you, go back to this here. Dichroic mirror is green. You see, this is transmission, longer wavelength. It comes down. Did you notice that this green is actually having overlapping with the red one here? Because dichroic mirror is not like ideal cut of 100% versus 0%. It has this slope there. So in general, I have to say, if you shine the excitation light to the dichroic, majority will be reflected as a mirror, but maybe about 5 to 10% actually pass through. What does it mean? Is when you shine the light, it goes down, and a lot of excitation light scatters, and part of it passes back to my eye, which makes a background. All right, so that's what you have to understand: presence of background fluorescence, and also not only the focus of the specimen. There's a focus in the specimen, but above and below there are still fluorophores. I want to see only this specific location or height, which I focus. But unfortunately, if the specimen is thick enough, then below and above, there's all fluorescence coming out, which is unfocused. That also blurs your image. So let's say Zui.
Yes, Professor.
Can you guess what this is?
This is our focus light.
Great. Yes, out of focus light. Okay. So that makes our microscope a little blurry. And for epifluorescence, why do we have to shine from the top, not passing through? Because the sample is usually thick. So if it's thicker, more than like several tens of microns, then you don't want to shine through in general. All right. So now I want to discuss a very difficult subject, but in a great laboratories and in good places, we will have a more sophisticated microscope. And this is a chance for you to understand what different kind of fluorescence microscope is. I have to say we have confocal microscope and we have two-photon microscope in our institution, okay? So this is a great example. If you look at A and B, can you see the differences? A is a little more hazy. You can see more backgrounds over here compared to B, okay? So that is as we see, wide field fluorescence microscope, okay? But to overcome this, we scientists invented a trick how to make this image instead of this, make it like this. That's called this confocal microscope and two-photon microscope. For this, I have to explain you a little bit in detail of imaging process, making an image with a lens. You remember the Dutch Antoine Leeuwenhoek made the first microscope in history with one lens. What does the lens do? Lens is just bending light, that's all, but in a careful way. So if you look at this one, this is a one lens, okay? This is the focus at the tip of the yellow one. Then the light coming from this point will come out, diverge. And this lens, what does the lens do? It bends the ray of the light. So it converges back to a point here. So that's an image. If you make these points along the two dimension, this is point. But what I want to notice is, what if the lights coming from out of focus, such as this dotted lines from here? Consider an extreme case. This dotted line is, let's say, sun. Sun is very far away. If it's very far, what does the light is almost parallel, and they will focus at the focal plane of the lens. So the location is farther from the lens, then the focus on the other side will be shorter. That's the point I want to make here. Okay. Why I want to describe this is because typical microscope has light coming from above, below, everywhere. So that's why we have a blurry haze background here. So let's say I'm using a laser and I send the light in a very precise way, this. So from the focus point, and it forms a focus over here. And why don't we just pass through this light, which passes through this focus, so that I make a hole over here, which is called confocal pinhole. Okay, then a lot of light can pass through, so I can detect it. Then what about the lights coming from below? You can see the dotted line, the below lights coming out, and they will make a focus actually ahead of the time here. Then it will diverge again, so it will dilute by the time it goes to this pinhole. So majority of the light will be blocked by this pinhole. So that's the key idea, to make only light coming from the focal plane can pass through. So this is the idea. I say this point is corresponding to this point. That's why we name it as a confocal. The focus is common. Okay, this is an invention. For the first time in human history, we create three-dimensional microscope. It wasn't that long time ago. The first invention was patented, filed in 1947 by Marvin Minsky, who is a MIT professor now, who was at the time postdoctoral fellow at Harvard. Okay, so now we have to consider this is confocal microscope, which allows us to make remove out of focus light, and by using confocal pinhole. But you can notice that the disadvantage of this approach, you are losing a lot of light. Okay, so that's the disadvantage. Now we have invented another type of microscope in 1991, and that's called nonlinear microscope. So far, all of these ones we call linear. Linear means, the input becomes twice, the output becomes twice, proportional, linearly proportional. But we use photons more wisely to make it as a two-photon nonlinear microscope. What does that mean is we send the laser light and we focus. Interestingly, only at the focus, the light will excite the fluorophore. And out of focus, no excitation is possible by use of nonlinearity of two photons coming into one point simultaneously. Okay, so what's good about that? That we know exactly the emission light will only coming from this point. Then do we have to care about out of focus light? No. Then do we need this confocal pinhole? No, because we know all the emission photons are always coming from this specific location. So that you remove pinhole, you always capture all the emission photons, so that you can have a better emission photon collection. And then how can you make an image? You just move around these points in 2D and 3D. So this is like a magic. So it happens like about exactly 30 years ago. Now many biology laboratories and neuroscience laboratories are depending on this new technology. All right, so now I'm gonna go a little bit further for confocal microscope. What I describe is only about point, but microscopic imaging is not about point. Point has to move around to make an image, and that 2D image, you can move in Z different part to create a volume. So that's why we call confocal, but in fact, most of the time it is confocal laser scanning. You have to scan the point so that to make a 2D or 3D images. What I just described, it is in here. You see laser light source, and you make the light going into the dichroic mirror, and you focus at this focal plane. Here it's described as a blue and red, which is above and below. Out of focus light path will always diverge at the confocal plane. So here confocal pinhole eliminates most of out of focus light and only allows this green focus, which is focal plane, and they will pass through this confocal pinhole. All right. And I have to say confocal is a one-photon microscope in comparison with two-photon, which I'll describe the next time. So here, what are the principles we learned? Hey, we can selectively collect light from very thin regions of specimen, which is on the focal plane, okay? And we use pinhole aperture to reject the out of focus fluorescence. And for making an image, we typically laser scanning and 3D reconstruction is necessary. So here, what I want to tell you is look at the detector. Hey, this detector is not camera at all. This detector is just 1 pixel sensor. Okay, it's a 1 pixel camera, if I say. Then you only get 1 point information at a time. That's why you need a scanning, 2D and 3D. And then you have to reconstruct with your, like, let's say MATLAB to make this point by point, you create an image or reconstruct. Okay, so because this confocal is a 1-photon, I'm gonna describe 2-photon microscope. Now, this is like 숨은 그림 찾기. This is like you find what are the difference between left and right. So let's go back to Seung-Young. So left is confocal, right is 2-photon. Can you describe what are the differences between these two?
The confocal microscope has pinhole.
Yes, pinhole. 조지훈?
Yeah.
Can you see the left and right? What are the other differences?
The focal plane is not. It's focused on the surface of the water.
You mean this one?
Yeah.
So it doesn't have these ones.
Yeah.
Okay, that's kind of right. Yeah. 최유진, can you see any difference left and right? I mean, you look at the color. Do you see the same color or?
Different color.
Excuse me?
Yes.
Yeah. Can you say it again?
They have different color of light.
Right, exactly. That's the point. All right. So you remember confocal is a 1-photon microscope. We shine blue light, okay? And then we get actually yellow light comes back, okay, which is a longer wavelength. But here you notice the shining light is not blue anymore, it's a red color, and then it comes back the same yellow fluorescence. This is a big difference, and when you design and build a two-photon microscope, you cannot use the same dichroic mirror because now you are shining actually longer wavelength than the shorter wavelength fluorescence. So this dichroic must have flipped somehow because it has to reflect longer wavelengths and transmit shorter wavelengths. So you need a specific different dichroic mirror for two-photon microscope. Okay, and Ji-Hoon marked very well that, hey, somehow the difference here is two-photon, you only have focal plane 1 point and no above and below because there's no fluorescence happening out of focus. That's the key part of two-photon microscope. All right, so I want to show you a real picture. So this is a sea of fluorophore. So whenever the excitation light, you see the light path. This is confocal. You see this kind of a wine glass shape because this is the path. And when you notice is fluorescence happens not only at the focus, but above and below. But in two-photon microscope, if you look at closer, can you see a point here? Can you see a tiny dot? Maybe not. Because of that, I have this inset. Really, there's a point. They only fluoresce. Okay? So you don't care out of focus. Why this happens? Because this is nonlinear microscope. And think about your basic knowledge of physics. Many things in life looks like linear, but in fact, it's everything's nonlinear. You just don't notice it much. However, when things are in specific consideration, for example, you focus light, let's say fluorescence happens when excitation photon gets onto the fluorophore, it excites the energy state and the energy goes back, it emits another emission photon, which is longer wave. However, if you shine the photon so high that the density or flux of photon excitation gets so high that there are chances that one photon and two photon simultaneously excite this molecule. Then how does the molecule feel? Actually, it feels like, oh, I've got two photons simultaneously, double the energy. So you don't need a very high energy. In fact, you need only about blue, which is longer wavelengths, but the effect is actually double the energy. So effect is similar to blue, okay? And that's the nonlinearity happens. And that's why it only happens, probability is at the focus and not out of focus. So this is nonlinear phenomena, and you need to have a special laser light source for this to happen, okay? But this is great and amazing that it looks like a magic. In 3D space, only 1 point. And you just move around this point laterally to create a plane image, and also ups and down, you create 3D or true 3D images. Compared to confocal, this confocal is a tricky 3D because you actually shine above and below. And what's bad about this? Because the sample you have, once it shines, it will photo bleed. The sample will degrade, all right? So confocal is a trick to make this semi 3D while you actually shine everywhere. So if you are interested in like time-lapse microscope, to keep the cell alive for a long time, you have to expect some things can go wrong because you are shining all the time, while two-photon is not the case. Okay, hopefully that makes you understand and you can now answer these principles. A fluorophore can be excited by effectively absorbing two photons simultaneously. And that fluorescence excitation restricted only at the focus, because this phenomenon can happen only at the focus, right? So this, let me go back again. Then what kind of interesting result out of this is you don't need to use a shorter wavelength, right? Bluer light. You can use a longer wavelength. And here I have to explain the light scattering and wavelengths. So why does the sky look blue? Even though you know the sunlight is like giving you all the rainbow color, which makes it whitish to our eye. Why the sky look blue? Because of the rainbow blue color, blue photons, which has shorter wavelengths, can interact with the Earth atmosphere's molecules, and they scatter down. They scatter better, so that's why we see blue photons compared to red photons, okay? So shorter wavelength scatters light more, and scattering is sometimes not good for our imaging because it blurs our images. So look at this two-photon, and why the sunset is red? Because of the photons coming from the sun passing through the thick atmosphere. The blue light actually scattered out, only red light remains to see our eye a beautiful, orange and reddish sunset. So red color can pass through longer, deeper. So this red color allows us to see deeper inside tissue. That is an amazing advantage of two-photon. So deeper imaging is possible. I mean about like around less than 1 millimeter. Confocal microscope, just remember confocal microscope, you can image only about 100 to 150 micron for real tissue, all right? While two-photon can go about 2-3 times deeper. So that's it. Why? Because we are using longer excitation wavelengths. And there's another reason. If I say 2 reasons why two-photon microscope can go deeper, is another reason is, remember confocal microscope use pinhole to collect light? That's okay for very thin cellular sample, but what about thick sample like brain slice or real in vivo brain? There's a lot of structures. The photons will even start from point. They will inevitably interact with other molecules and change their path. So the ideal diagram, this doesn't hold because of scattering. So you actually lose a lot of light even from the focus if you use confocal pinhole. So why two-photon can go deeper? You don't need a pinhole. You just collect all the emission photons. So no pinhole for detection. These 2 tricks allows us to actually see deeper inside the brain. And that's why this becomes the workhorse for many neuroscientists who want to use microscopy to understand how neurons and dendrites and synapses are changing, okay? So another advantage, like accompanied by this principle, is actually less light scattering and less photo bleaching. Confocal has above and below fluorescence, two-photon do not. So it doesn't have as much photo bleaching, and also phototoxicity, which is coming from making fluorescence. The cells inevitably generate more toxicity, okay? Reactive oxygen species, and those are lesser, so less phototoxicity for two-photon. So how can you use this one? This is good for extended observation of cells in living brain tissue in whole animals, or fine-scale neural structures in brain slice. So I hope you understand the distinction between these. But nothing is for free, and two-photon is expensive. Why? To make this nonlinear microscope, you need a special laser. That's very expensive, okay? And the equipment is, of course, for two-photon. The lenses are more expensive, so there are limitations. Okay, so why are we talking about this? Because of we want to see inside the brain. So two-photon, this is a Jablonski diagram, which you may take later on more advanced course. I'll just give you the concept. The length, the arrow length means energy. So two-photon have simultaneous this of excitation photon coming. So that's why double the energy. And then they lose their energy in this excited state. And then they come down to emit a photon which is proportional to this length or energy. So that's two-photon fluorescence. And you guys are supposed to do brain surgery to create a cranial window at the end of the class. So let's assume this is the case. You take off the skull bone and you put the window, okay, glass window, and that allows you to be able to see inside the brain. Especially two-photon, you can see real inside the brain. You look at this green fluorescence coming up. And there are another advantages of two-photon microscope that you can see simultaneous multicolor imaging is possible. So look at this. This is a staining for blood plasma. You cannot see this without injecting some dye into the bloodstream. So in this case, we inject the dye so that you can reveal the shape and the function of blood vessel. And in this case, this left one, also we will discuss more. We can actually give a calcium indicator to neurons. So that neuron will show green fluorescence and they actually blink because neuronal activity comes with this calcium ion flux or changes so that you can dynamically see this. And this is an example of this specific dyes which show the neuronal structure and surrounding blood vessels. And even for those who are studying Alzheimer's disease, there's even dye which makes this amyloid plaque to show specific color so that you can actually see and quantify and analyze the amount of amyloid beta. And another very interesting part is neurovasculature, because many of the modern diseases are related to neurovascular origin. So you want to study vasculature, and here you can actually have this fluorescence coming specifically from smooth muscle cells that wraps up around the artery. Not the vein, but artery has this because you have to control this blood flow. We know that our brain has only 2% of our body weight, but it spends 20% of all our oxygen all the time, and it spends 25% of glucose resource in the brain. So you need to have an exquisite control of where to send this blood, oxygenated and nutrient blood. So you need to have a very good control of this sending the brain location, distribution of blood. And that's a surrogate marker for neural activity. That's how we use functional MRI to be able to tell which part of the brain is activated when you do specific neural activity. Okay, so TILF, this is a fascinating subject, but I have to skip. I just want to tell you one point, which is when you want to study a very fine structure, which is happening at the surface, near the surface, we actually can create a light sheet, which is only present in very thin layer here, so that you can excite the fluorophore here as a red dot, not these green ones. So this allows a complete blockage of black background, and you can watch what's happening here. This you can consider as a synapse, synaptic cleft, and those ones are very tiny phenomenon, and you can use this one. So what I can say is You know, Nobel Prize winning super resolution microscope uses a lot this total internal reflection geometry. And in the life science, using imaging, majority of single molecule studies are using TIR geometry. And TIR means the incident light is so high that it won't get into the sample, but it reflects it back. And only some light remaining are present here. We use that light for imaging. Okay, so this I put as a reference, but I do ask you to at least try to understand this lab too. Excitation light, and then dichroic mirror and emission. Focus on the shape of the excitation light. Epifluorescence will shine through the sample, while, you know, this is a inverted geometry, so that you can have a cell culture, you open up the space, you can have a patch clamping, you can have electrophysiology from the top. So that's why we use inverted microscope. But look at confocal, the excitation light actually focuses down inside the sample. Compare the difference. And you use this point left and right to create an image. So that's the difference. And epifluorescence, you use actual imager, which is camera. But confocal, you use a point detector. In this case, APD stands for avalanche photodiode. This is just a one point detector, very sensitive. Or you can use photo multiplier tube, which is called PMT. Okay, so let's see. Before getting into electron microscope, I'm gonna answer one of the questions by Kohiner. Is there any limitation how deeper we can see by a two-photon microscope? Very good question. Is it depends on the energy of the emitted photons? Yes. If we can increase the energy of the emitted photon, can we see deeper? So the energy of the emitted photon is in its color. So if the energy is actually lower, then it has a longer wavelength, we can see deeper. So the depth of imaging depends on the color. And limitation, how much deeper, why we cannot see below 1 millimeter. This is a subject for complete other like course, but what I can say to you is the photons, when it gets interact with the tissue, it scatters. Because of this scattering, we can form an image, but also out of focus scattering makes the imaging blurry. Because of that, we cannot see throughout very deep nature. But a very up-to-date study actually tried to overcome this limitation, which is also a fascinating subject. That's what I can tell you right now. Okay, let's move on to electron microscope. So let me ask a question to Shukla. Left and right, as I said, there are two kinds of EM, which is scanning EM versus transmission EM. Which one is a scanning EM? Can you tell? Left or right?
This is a scan. Second one is a scanning.
Yeah, I'm asking you, which one is a scanning electron microscope?
Second one, 10 micrometer.
Okay, that's fine. So this is the question. After this, you guys should be able to tell, hey, the right side one, this is transmission EM. Left side one is scanning EM. Scanning EM looks like 3D. Transmission EM looks like a slice, okay? This one has a better resolution than scanning EM. Okay, that's what you have to see. So, okay, why electron microscope? Because of the limitation of resolution in light microscope. As I said, when we use light for a mean to resolve something in imaging, we depend on the light wavelength, which is especially visible range 400 to 700 nanometer. You have to cut it in half. Half of the wavelength is the kind of a rule of thumb. So you can have about 200 nanometer due to the wavelength of the light. But think about the object of this course. We are interested in neuron. We are interested in neuronal synapse. We are interested in synaptic vesicles. That's a 30 nanometer. We are interested in protein, which is below nanometer. We can't see that with the light microscope. So sometimes we have to use another tool. So the concept of electron microscope is we have to use a wave. But you know, the famous De Broglie in physics, who got Nobel Prize by proposing even the particle of electron have a wave phenomenon. We call it metal wave. Electron is a particle, but that's so tiny that has its own wave. And that wavelength is tiny, 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 so that we can utilize the wave nature of electron to make a microscope. So that's the key idea. So what's great, you can have even sub-nanometer resolution, okay? So that you can observe synapses, synaptic vesicles, and even ion channels. Wow, that's great. You know, can we see this? But you have to pay cost. Nothing's for free. To make this possible, you have to kill the sample. You can't use live sample, all right? So specimen must be fixed and dehydrated and chemically treated, okay? The sample looks like black stuff because of the osmium, a toxic chemical which prepares the electron microscope. So you can't do live sample. And think about while during this harsh fixation, dehydration, the sample will inevitably change its morphology, its original structure. So that's an artifact. So you have to keep in mind potential artifacts from this processing of the sample. And two category, which I asked Shukla. Sorry, this is a little bit difficult question initially, but the point is this sample, EM, you have to cut the sample like super thin. I'm talking about several tens of nanometer thickness. And then you shine electron, and transmission mode, you take a picture. That's we call TEM. That's why the sample looks almost like slice. So if you look at something like this, that's TEM. On the other hand, scanning EM is you focus electron shot to a point, like confocal microscope. You have a point shooting. You keep shooting while scanning, and then you collect the photon. Look at that. It looks almost like three-dimensional. But to make this, you can't use the sample alone. You have to coat the sample on the surface. So scanning EM, you're not seeing actually inside the sample. You're only seeing outside. That's the point. All right. So let's compare light microscope and EM. So this is a hair cell structure, and A is a light microscope. Can you see something like this, like shady shape? This is DIC microscope, differential interference contrast microscope. And this one, if you use SEM, scanning EM, wow, it looks like great detail here. Okay, it looks like 3D. But you cut this one into a slice and you use even higher resolution of TEM. Transmission EM will give this detail. So that's the comparison. And I want to show, hey, then how does the electron microscope, how can you build this? In fact, light microscope use photon. To bend the photon, you do need a lens. How to bend the electron? Electron has an electricity, okay, charge. So instead of a lens, you use a coil or electric field to bend the electron trajectory. So this is light and this is electron microscope. It looks very similar, but how to bend? There's a lens, there's a coil. There's a lens, there's a coil. Does it make sense? So that's the electron microscope. Typically, this is a little complicated, so it's more expensive. So here comes then, what about TEM versus SEM? All right. So then light microscope, now you guys are experts in light microscope. Let's say we are watching like a sperm cells, let's say human sperms. You can have a live image of sperms like moving around, swimming around, but you can't do it in electron, but with a high resolution. Look at this sperm head, you cut it, and then TEM shows much more detail inside the structure of the sperm head. How? By using transmission EM. How to prepare that? You have to cut the sample so the electron can pass through with interacting and scattered from the sample. So here, your specimens here, the electron has to pass through, and the rest is almost like light microscope, but you need to watch with the eye. So there's a fluorescence screen, which is making the electron bombardment will convert into a fluorescent light so that you can actually see or you can actually record. What about SEM? SEM is a little different. You have to shine the sample with a focus. So, and then you have to scan. So what you need is here scanning electromagnet. So you use electromagnet to deflect the electron beams X and Y. So throughout the sample, you go through this raster scanning, and you do have a detector to collect the scattered not photon electrons. So that this is how you make scanning EM. Hopefully this makes sense. Then TEM is a thin tissue section. TEM is thus 2D images, okay? Electron beam is aimed through a thin, it has to be thin. So it's a tens of nanometer, less than 100 nanometer for the electron to pass through to section of a specimen that has been chemically treated to enhance contrast. This is important, chemically treated. You are not seeing the sample directly, the sample molecule. You are seeing that treatment, which is specifically attached to the structure of the sample. Tissues are stained with atom. Hey, electron has to scatter, then it has a high energy. So you need to have another high energy on the sample. So you put the atoms of heavy metal, which is electron dense material, you mix with the sample and you're watching that, which is preferentially attracted to certain cellular components. So electron microscope is not specific, you have to understand, not like fluorescence, all right? And then instead of lens, they use electromagnets to focus and magnify the image by bending charged electron trajectories. In fact, it's the same. Light, you are using high refractive index lens to bend. Here, you are using electromagnets. And you're projecting electrons into a special detector or onto a fluorescent screen to form an image. What about SEM, scanning EM? Useful for detailed study of specimens. SEM is watching only at the surface, okay? A high energy electron beam scans across the surface of specimen, and that specimen, you can't use just typical specimen. You know, I prepared some EM. You have to go to cover with a gold, okay, expensive gold coating. Because of that, you can actually take an image, okay? So high energy electron beam scans across the surface of specimen, usually coated with a thin film of gold or platinum, which is also, of course, to improve contrast and signal to noise ratio. And that emitted, not photon, but electron signal will be collected, processed, and constructed as an image of surface topography that appears as, in fact, you are not seeing inside, but only cover, but that looks like a 3D. Okay. So now I have to say the more advanced part, which is electron tomography. How can we see inside? That's a question. Of course, this is more recent development of electron microscope, and now we can have electron tomography, which is, look at this. This is tiny synaptic vesicles. You know, that's like 30 nanometer size, which is impossible with light microscope most of the time. But tomography sees inside. How? The specimen is tilted. Hey, there's a specimen. You make a one transmission EM, but the specimen has actually thin, but it has a structure. And you tilt this and make an image. Tilt this, make an image. Remember, the CT, computed tomography, is actually shining the object with different angle and to reconstruct 3D. Exactly the same concept. TEM, your sample here, shine, you tilt, different angle, and then you compose, reconstruct with these 2D projection images to create not exactly, but almost good enough. So tilted within the EM to produce TEM, not SEM, many different perspectives or to reconstruct a 3D view. Of course, with this, you are losing a little bit of resolution because you have to use a little thick sample and your resolution is about nanometer order. Why do we use this? Look at this, a nice interaction of synaptic vesicles at the cell surface. This is just an amazing. You will see this as a cartoon in your textbook now, but this is a real image, okay? For determining molecular structure and 3D, when you use EM, we are talking ultrastructure, okay? Because we are seeing way higher resolution than conventional microscope of an organelles. The organization of presynaptic vesicles here in axon terminal, we are watching axon terminal presynaptic vesicles. So that's now possible. So this is an amazing era that we are be able to see an exquisite molecular detail of things happening.